Okay, let's get started. So the answers are all over the place. So let's take a look at this. All right, so let's do a little bit of review. So this is a saturation recovery sequence, which means we tip the spins from equilibrium magnetization 90 degrees. So at the beginning, mxy is equal to the full magnetization, and m sub z equals 0. Okay, so saturation is when we start off at mz equals 0. So it's recovering from 0. Okay, and from last time we know that the expression is the intensity is going to be the density weighting, okay, times the T1 recovery times the T2 decay. Okay, so that's what we have for the expression. So let's take a look at each of these things. And the tricky part here, which I realized looking at the answers, is we said that a rule of thumb is, you want to take the average of the T1s. But what happens when there's 3 T1s? Okay, what happens when one of those T1s is way out in left field? Okay, so it turns out in that case, you don't necessarily want to take the average, right? You want to take the average of the two that are closest together to differentiate those. The other one's going to be very different from those anyways, and so you're going to get the contrast you want. So keeping that in mind, let's look at the T1 weighted image. Okay, so for the T1 weighted image, which have the two closest T1s? Gray and white. So what's the average T1 of gray and white? About 1100, okay? So for this case, we'd want to pick TR equal 1100. Okay. And then what should the echo time be? Ideally, it'd be 0. Okay. In practice, it's probably a few milliseconds. And let's do a sanity check. If I make the TR equal to 1100, does that make the CSF dark? Okay, so the CSF has a T1 of 4,000 milliseconds. So is it going to be, so, and here you notice on this image, it does appear dark, right? All this area, the cerebral, the ventricles are the middle part. Now let me just, so a bit of anatomy, okay? So this is the CSF. Okay, so these are your ventricles. Those are the fluid cavities in your brain. This part is the white matter, so that's all the wiring in your brain, which we'll look at later. And then the gray part here on this image is the gray matter. That's all your neurons. That's doing all the calculations. Okay, so that's for just anatomy in 30 seconds. All right. So in this T1 weighted image, the CSF does appear to be dark. So does that match with, if I use a TR of 1100, does that make sense that the CSF will also be dark? Okay, that sounds like me. How are you getting that? Previous lecture. Okay. Yeah, it does make sense because if I'm only waiting 1100 milliseconds for the signal to recover, right, and the time constant is 4,000, it means I haven't gotten very far on the recovery. So it's going to appear fairly dark. Okay? So with that in mind, any questions on T1 weighted imaging? And this is just one kind of T1 weighted image, sort of the simplest case. There's much more sophisticated kinds of T1 weighting involving many more pulses, but this is the simplest kind. Okay? So the basic idea is we are picking the TR to differentiate gray and white, and by doing that, it also happens that the CSF also appears dark. Okay. All right. Let's take part B, density weighted. This should be the easiest one. So what should the TR be here? I want to make this term go away, right? This term here, I'd like this term to go away for density weighting. So how do I make that term go away? What should the TR be? Just go ahead. Really big. Okay. So we could make it infinity, right? But that's not a really good answer. So what would be a practical answer? How long should we wait, which is almost as good as waiting infinitely long? Several times the time constant of what? Okay, so you have three choices: CSF, white, or gray or white. So which one should we make 3 times? CSF. It's the longest one. So if we want everything to recover, we need to pick the longest one, wait for that to recover. So the TR will be 12,000 milliseconds is a good sort of rule of thumb, right? By 3 time constants, most things have recovered, and you don't necessarily need to wait. By waiting another second, you're not going to get much more, okay? So that's a good rule of thumb. And then TE, want to make it 0, right? Because that will make this term go away, okay? Let's look at the last one. So TR should be what? I want T2 waiting, so I want no T1 thing. So TR should be 12,000, right? And what should the echo time be? The average of what two numbers? Yeah, the average of the T2 in gray and white, which is what? It's about 95, I guess. Is that right? 95? Okay. Okay, everyone okay with that? Okay, any questions on that? That means you're all ready for the next question, right? Okay, perfect. All right, so this was actually on last year's quiz 2. So if you're okay with this one, you should be okay with quiz 2. If not, we'll go over it. So go ahead and take a look at that. And I think this one is just a regular old poll. So let me activate that. Okay, so that should be ready for you to just put in your answer. So it's a multiple choice, so just, you feel free to discuss it, but at the end of the day, just each group just enter in your best answer. Okay, about another minute. Go ahead and submit your best guess in the next minute. Okay, let's take a look at this. Things are still changing quite a bit, but it's clear the two most popular answers are C and D. So this got 39%, and this has 48%. Okay? So let's take a look at So, and these are basically diametrically opposite answers, right? I mean, so it's like we're at chance now, right? Okay. So let's take a look at this and see if we can figure out which one of those two it should be, or maybe it's A and B, okay? So, so you've got these liquids. They've got T2s of 1,000 and 2,000, right? And you add some gelatin to liquid A, and you want to keep the contrast approximately the same, okay, between these two liquids. So what's going to happen to the T2 of liquid A when you add the gelatin? It's going to get longer or shorter? Shorter. So why will it get shorter? Anybody? Right. So there's what's happening to the tumbling rate as we put the gel in? It's less tumbling, right? Okay. So remember this graph here of tumbling rate versus T2? Okay, it sort of goes like that, right? The more tumbling there is, the more motional averaging, right? And therefore, when we're in this regime, the spins don't really affect each other because it's just averaged out to 0. As we move down this curve, which we're doing with the gel, we expect the T2 to go down. All right? So we expect T2 to go down, and therefore we expect the average echo time should also go down because the average of the T's has to go down, right? So the average T2 should also go down, and therefore we should make the echo time go down. Okay, so that means it could be either C or B. Okay, so that rules out, sorry, B or D. It can't be A or C, right? Everyone okay with that? Okay, so now we just have to choose between B and D. So do we want to increase the TR or decrease the TR? And it's a forced choice here. It's not letting you keep the TR the same. So what would happen if we decrease the TR? What might happen? The what? Yeah, we might introduce some T1 weighting, right? So that's probably not good because it doesn't say how much we're going to decrease the TR. If we could decrease the TR a lot, then it's going to obviously introduce some T1 weighting, right? So the correct answer then is D. Okay. Any questions on that? Okay. So the quiz 2, we're still finalizing it, but we're almost finalized quiz 2. It's going to have mostly multiple choice and a few free response, but the idea is you're going to have to try to do each multiple choice in about 5 minutes in order for you to get through all of them. So you want to we'll go over some of them after today, and I'll try to record that as well. But that's sort of the type of question you'll get on the multiple choice. All right. Anything before we continue? Okay. And that's next Wednesday. Yeah. Okay. So far we've talked about saturation recovery, where we tip the spins 90 degrees. Okay. And so we had this saturation recovery idea where we tip, so the M sub Z component went to 0, and then we had to wait around all this time for the signal to recover. Okay. So it turns out that waiting around is not necessarily great because time is money. It costs, every minute on the MRI system costs money. And then even if you have the money, oftentimes the patient may not be able to lie on the bed for that long. If they've just had an emergency, you might be very limited in time. You're trying to get them in and out as quickly as possible, trying to get, especially in the ER situation, you really want to sort of minimize how long you have them in. If someone's in a car crash, you're not going to have them in there for 2 hours. Okay. If they're part of a research study, yeah, maybe 2 hours. But for an emergency situation, that's time is critical. Okay. In the stroke business, time is brain. Okay. The longer you wait, the more brain you lose. All right. So timing is very, very critical in the clinical environment. So one question is, why do you have to tip it 90? Why not just tip it less? So for example, this is the theta, the tipping angle. Why not pick other values of theta And therefore, you haven't, instead of starting off from 0, you might be starting off some other point halfway through, and then you're much closer to where you want to recover to. Okay? So this was the insight that a physicist, Axel Haas, had in I think it was the late 70s or mid 80s. It made him a really rich guy because he patented this idea. Okay? And the idea was that one way of thinking about this is what's called the toilet flush analogy, right? So those of you, you know what a toilet tank looks like, right? And it has to fill up, right? So the idea is if you, a 90-degree pulse is like flushing all the water out of the toilet. And then you have to wait around for the toilet to refill. So that might take, a minute or something, or 30 seconds, okay? So that's the T1 recovery of your toilet tank. So the idea is, what if you just flush the tank halfway, right? You might get almost as good of a flush, right? And yet you don't have to wait as long to do the next flush. And then you could just keep flushing it and flushing it, right? Okay? So that's one way to sort of think about how the flash sequence works. It makes this trade-off between maybe you're not going to get a full flush, but you get enough that you don't have to wait around as long. Okay? So it turns out that is actually true. And so what's called the flash sequence, or the low flip angle sequence, is used a lot in clinical imaging to speed up. Especially at the beginning of every MRI scan, we do what are called localizer scans, where we just want to figure out where's the body and sort of do a scout scan. And then we prescribe the additional scans on those localizers or scout scans. So we want to make those scouts as quick as possible. Okay, so typically those only last for maybe 30 seconds at most. All right? And those are done with flash sequences. And so what they realized is that you could have this trade-off, and that you could also pick a specific angle called the Ernst angle, which, given a TR and a T1, it tells you the exact angle to use that will maximize your signal. So that's shown here, where we've assumed that the T1 here is 1,000 milliseconds. We pick 3 different TRs, and you can see for each curve, as a function of flip angle, there is an optimal flip angle, okay, which is named the Ernst angle after Richard Ernst. And so for comparison, these are the values you would get with saturation recovery. So if you just did the typical 90-degree and waited around at different TRs, so this would be different TRs, you would be picking up the signal at different TRs. And so you can sort of see if you do saturation recovery with TR of 30 milliseconds, you only get 0.030, whereas if you use the flash, you get like 0.13 or something. So it's like a factor of 4 increase. That's huge, right? Similarly, at TR of 100 milliseconds, using saturation recovery, it's only 0.090. Here you get like 0.22, so more than a factor of 2. As the TR gets longer, the gain is less. So here you get 0.39 with the old method and maybe 0.49 with the new method. So as the TR gets longer, as you'd expect, the two methods converge because you're waiting around a long time. But the key is if you pick a small flip angle and you're not going to wait very long, so 30 milliseconds is not very long, the flash way outperforms the saturation recovery. And so that's why it's used for very fast imaging. All right, so that's just something that you should know just because that's used a lot. All right, any questions? The other one I want to talk about is something called inversion recovery, because that's also used quite a lot in the clinical practice. And the idea here is, so far we've looked at this part of the pulse sequence. Okay, we had a 90-degree pulse. The 180 was to sort of rephase the spins, and then we acquire the data here. Okay, and so we have this timing of echo time was the time from the 90 to the middle of that acquisition where kx was equal to 0. Okay, so that's what we defined as the echo time. Now what we're saying is before we apply the 90, we're going to apply this 180-degree pulse, and this is going to be called an inversion pulse. Okay, and what it does is, if the magnetization is here, it just inverts it and makes it point in the opposite direction. Okay, So going from m naught to minus m naught. And so now the recovery starts at minus m naught. It's trying to go back to m naught. And you notice it has to go through 0. And that's the key. We're going to say, okay, that's really useful. That means we can get rid of signals we don't want, right? If we time things correctly, we can zero out signals if we acquire at this point in time, all right? And it turns out that this TI is called the inversion time. It's the time between this inversion pulse and the 90-degree pulse, okay? And it's given by this equation here. So if I know the T1 and I have some TR, I can calculate what that inversion time should be. All right. So in the limit, as TR goes to infinity, this term is going to go to 0, and I'm just going to get inversion time equals 0.693 times T1. All right. So that's just a nice to know that you can use in MR, you can actually null out tissue that you don't want to see. So this is an example here where we've done selective nulling of different things. So here, it looks like here we've nulled out. So depending on where you acquire, so for example here, if I acquire here, I null out fat. If I acquire here, I null out white matter. And if I null out here, I null out cerebrospinal fluid. All right. So this is an example of nulling out fat. And this one here is an example of nulling out CSF. So you can sort of see the CSF here is very dark. Okay. And then for here, I've nulled out white matter. So sort of see all this stuff here that was white on the T1 weighted image. Now it's all very dark. Okay. And then here, which is not shown here, this one here, I've nulled out gray matter. So that's a sort of interesting looking image. So basically all the neurons there, their signals are nulled out here. And so you see the white matter tracks here, but all this stuff here where there is brain tissue, we've nulled out the signal. All right? So, so this is how it's done. Basically, it's essentially like any sequence you would have, but you just apply a 180-degree pulse. Okay, and that's the inversion pulse that you apply. And so in this case, we're nulling out fat. In this case, depending on where we acquire, if we acquire here, we're nulling out fluid. And you can sort of see the fat and the muscle have both recovered to reasonable values. So here we would see the image, we'd see the fat and the muscle, but the fluid would be black. Okay. Here, we'd null out the fat. The muscle and the fluid are negative, okay, but when we flip a negative onto the axis, there's still signal, and so we would still see the muscle and fluid there. Okay. This is a pretty interesting one. This is called black blood preparation. It's used a lot for cardiac imaging, where you want to sort of get rid of the signal from blood that's in the heart, so you can sort of see the heart wall better. And so this is interesting because you see here they apply two 180 pulses. Here, non-selective and selective. So the non-selective, and we'll talk about this on Wednesday, just inverts the blood everywhere. Okay, so it inverts for fat, muscle, and blood. They all go minus. And then what it does is selectively it applies another 180 only to the stuff that's in the heart, and it leaves the blood outside alone. And so that applying a 180 pulse to something that's inverted just flips it back. Okay, so here you see the fat and the muscle have been flipped back, but the blood is still negative and is recovering. So at this point, when you acquire the image, you have blood black and fat and muscle pretty bright. Okay. So this is an example here of, this is sort of a flare image where we've nulled out CSF. And here, this is a black blood image. Okay? And the reason to do that is if you didn't do that, there'd be a lot of signal here, which would make it harder to see that vessel wall. And oftentimes you want to be able to just look at that vessel wall very clearly. Right? So that's one example. So inversion recovery is used a lot clinically, and so it's good to know about that. The other one that's starting to be more clinically, but we've been doing research in this area for, let's say, the past couple of decades, is something called arterial spin labeling. I'm just going to just present it as an interesting way of talking about what happens when you invert spins that are moving. So later on, probably in this lecture, we'll talk about moving spins. But the idea is that in this case, we apply the inversion pulse down here in the neck as the blood's flowing up the carotid arteries. Okay? And so these spins, you imagine I've tipped the spins in the neck, and they start flowing up into my brain. And as they flow up, they also recover. Okay? Now, if I acquire the image before they fully recover, then I'm not quite back at m sub naught. Okay? So I have some signal it hasn't quite recovered. And then that's called my tag image. And then I acquire another image, which I call my control, where I just let the blood just flow up relaxed. Okay? So when it gets there, it's still fully relaxed. Okay? So you can sort of see there's a difference between these two signals, right? This signal here on the bottom is going to be larger than this signal here, assuming the blood has not fully relaxed. So the size of that signal is going to be proportional to how many spins went from here to here. Okay? And that's going to tell me about cerebral blood flow, which is pretty important. We all need blood going to our brains, right? So this is a different picture of that. So the tag image, we invert it here, and it recovers. And then the control image is fully relaxed all the time. So this difference here is a measure of how much blood actually got there. Okay? So what it looks like is we might tag the blood, we might acquire it, and then the next round we control, acquire, and then we just repeat that process over and over again. And it turns out if you do that, remarkably, you end up with these pictures. So these are very pictures that are about 5 minutes. And the nice thing is there's no contrast involved. It's fully non-invasive. We just tip the spin in the blood, and you can have these very nice images of how much blood is getting to every part of the brain. A colleague of mine, Eric Wong, also invented a method where you can actually figure out each of these colors represents how much blood was delivered by each different artery. Okay? So here you have your carotids and your vertebrals, and they're all delivering blood to different areas. And that's something you might need to know about if there's a stroke, for example, or there's an occlusion of one artery. You'd like to know how is the blood flow distributed. Okay? So this is an example of someone who had severe stenosis of their internal carotid artery. And so you can sort of see in this case, here, before they got a stent, this whole side here is, because it's blocked by, the carotid is blocked, is not getting any blood from there. So all that region is being supplied by their posterior arteries, which should only be, ideally should only be supplying the back of the brain, but now they're supplying the whole side of the brain. Okay? And then what happens is they put a stent in this person, and then afterwards you sort of see this region now, they've recovered the flow in terms of now the artery, the carotid artery is supplying the blood as it should. Okay? So it's very useful if you've got any occlusion of the arteries, and so there's a lot of ongoing research being done around the world, including here at UCSD, especially for stroke. Okay. Any questions? All right. So now we're going to switch to a specific kind of contrast, which is what my area of research is, which is FMRI, okay, which stands for functional MRI. And so far we've really been talking about brain anatomy and sort of how do things look. So we're going to spend a few minutes talking about what does the brain actually do and how does it function, and how can we use MRI to study how the brain functions. All right? So an fMRI experiment is very similar to any MRI experiment in the sense that you still put the person in the magnet and they lie there. The main difference, though, is in a lot of fMRI, you actually add some stimulus. They might be looking at a screen, or you might be piping in different smells or taste, or zapping them, or doing lots of different things to them. Okay? Heat pain, you might be heating them up and causing them pain. You might be pricking them with acupuncture needles, just lots of things, all right? And then they also have the opportunity to respond. So they might be gambling or making decisions or trying to track some object. And so there's the main thing that makes it different from just MRI is the subject. You're asking the subject to do something. So fMRI as a field is actually fairly young. It was really, in humans, it really wasn't until the early 90s that it took off, and it was shown that you could do it in humans. And so this is from one of the first papers where essentially what they did was they put some humans in a magnet and they shone light at different points in time. So the people are lying there, their eyes are open, it's either dark or light, dark or light. And what they found was that if they looked in this posterior part of the brain, which is where your visual cortex is, they could actually detect these signals that went up and down and up and down. Okay, and so this was the first proof that you could actually measure function non-invasively with MRI in humans. And so we're going to talk a little bit about this. How does this work, and what does it mean? Okay. So as I said, fMRI is relatively new. It didn't start until the early 90s, but has quickly become sort of the predominant way of measuring brain function in humans. And the main reason for that is you don't have to inject any contrast agent. Okay, before that, PET and SPECT, these two modalities were the main way to get brain images. Okay, so before the early 90s, this is what you would do, but it was very expensive and also required you to inject contrast agents in the subjects. With fMRI, all of a sudden you could just recruit normal subjects and within minutes just put them in the scanner, and there was no need for contrast agent. So that really changed the whole game. So one key, though, is if we want to study the brain, we have to make some trade-off, right? We can't, so for example, this is an anatomical image here on the left. It's called MP-RAGE, that's just the name of the sequence. And this took about 6 minutes to acquire a fairly high resolution image of the brain, so 1 millimeter resolution. Now, 6 minutes is okay, but the brain does a lot in 6 minutes, right? I mean, I challenge any of you to think about what you were doing 6 minutes ago precisely. You could maybe take a guess. I mean, obviously you're in this room, but what you were actually doing is hard to say. So what we want to do is be able to acquire images very quickly. And so, for example, this slice here was acquired in 60 milliseconds, and here the voxel volume obviously is much greater, so this is about probably 3.5 millimeter isotropic or something. So we've gone up in voxel size, but yet now with latest methods we can go through the whole brain in about a second. So every second we can go through the brain. All right. And the way we do that is primarily with something called echoplanar imaging, which you've seen on your homework and in previous lectures. So it's this zigzag sequence that we talked about. Okay. And typically the center of K space is acquired in the middle, and so this is where we're going to measure the echo time. And so typically that will be about halfway through the sequence. Okay? And so we're going to find out that setting the echo time for fMRI is really important. We don't want it too short, we want it too long. If we set it just right, we get these really beautiful functional MRI images. So the basic idea behind an fMRI experiment is you acquire these images very quickly, but at fairly low resolution. Okay, maybe 2 or 3 millimeters resolution. And then while the subject is lying there, you might present them with a task. So in this case, they're seeing a flashing checkerboard for 20 seconds, and then a blank screen for 60 seconds, and then you typically repeat that over and over again. All right? And then if you did that, you would get this response in certain parts of the brain that do that function. So in this case, it's a visual stimulus, so you're mostly going to get the response in the visual cortex. And then what you do is you apply a bunch of statistics, which I think one of the groups is going to talk to you about, and you come up with a map like this, okay, showing where in the brain did the time course look like the stimulus. All right? So you can imagine a very simple way to do that. You just do a correlation analysis, right? You just correlate that time course that you measured with your stimulus and then plot the correlation coefficient across the brain. And that's essentially what we do. All right? So this is an example of a finger, someone tapping their finger. You see activation here in their left motor cortex. So which hand were they tapping? Their right hand. Okay? So you've just done a very preliminary brain reading exercise. So from this, you knew exactly what they were doing. All right. So we want to talk a little about, so BOLD stands for blood oxygenation level dependent. Okay. So in MRI and FMRI, as in most areas of science, coming up with the acronym is a really big part of science. So BOLD, I'm sure they spent a lot of time figuring that out. Okay. But it's a good acronym. Okay. So anyway, this is BOLD contrast. And the main thing I want to point out on this slide is the dependence on echo time. So if I acquire t equals 8 milliseconds, I don't really have much signal, right? And in fact, in this exercise, in this study, when they moved the echo time out to 40 milliseconds, all of a sudden they're recovering this signal that they want to look at. So we want to spend a few minutes talking about why is there this echo time dependence of the BOLD signal. All right. So remember that we talked about T2 star, okay? And that if we have T2 star recovery, then the signal basically decays exponentially with T2 star time constant. So let's just review. We've already seen this slide before, but now we're going to imagine that this is sort of a cross section of a blood vessel, let's say, and just to sort of where we're going, this is going to have some deoxyhemoglobin in it. Okay, and in deoxyhemoglobin, at the center of the molecule, there's iron. Okay, so there's a magnetic substance that's flowing in you all the time. And the idea is if there's some deoxyhemoglobin in there that's a magnetic substance within the presence of this big magnetic field, so it's going to perturb the magnetic field around it, right? So that's going to lead to these field homogeneities around the blood vessels. Okay, so if I look at different spins at different locations, they are going to undergo different types of dephasing because they're experiencing different fields. So let's say they start off at time equals 0, they're all in phase. I wait some amount of time and they get out of phase. Okay. If I acquire at this point in time, the echo time, if I wait long enough, then they're out of dephase enough. Okay? And I can sort of measure how much they've dephased. If I measure too soon, like let's say I measure right up here, then I really haven't let enough time for them dephase. So I'm not really going to get much signal contrast. So I do have to wait some amount of time for this dephasing to occur. Okay. So now this is a case where let's assume that the deoxyhemoglobin level has gone up. Right, so there's going to be more iron here, and so you can sort of see here there's a lot more dephasing of a lot more perturbation of the field. So there's going to be much more dephasing, and therefore when I wait the same echo time, you see here on the bottom there's a lot more dephasing going on. And so I look at the vector sum here on top. This is the length of the vector I get, and then here it's much shorter. Okay, so that means if I have this sequence, I have the MRI system is actually sensitive to how much deoxyhemoglobin there is in the blood. Okay, so that means that I have some way of measuring non-invasively that parameter. All right. So just to remind you, T2 star is it's equal to this quantity here. So T2 star is here, and we also call the inverse of that R2 star, which is the relaxation rate. Okay, so this is the time constant, and this is the relaxation rate. All right. And it's composed of both T2 and T2 prime. And both of these contribute to FMRI, but predominantly for most FMRI, we're going to use the T2 prime. Okay. So let's take a look at what things look like. So typically what happens in FMRI is we'll measure some signal during a baseline state, like a blank screen. And so things are decaying with a rate of R2 star baseline. And so for example, in this case, it's this curve here. And then during the active state, we find that the relaxation rate changes. All right. And just empirically, for most studies, most parts of the brain, not all of them, but oftentimes we see a signal increase. So we go to this curve here. So this curve here is decaying with R2 star baseline, and this curve here is decaying with R2 star activation. Okay? And so because this curve, the red curve, decays more slowly than the green curve, the difference between activation here, so this is activation, and baseline, that A minus B is going to be positive. And that's typically what we see in a lot of FMRI. Not always, but for now, let's just assume that that's the case. Okay. So for that to happen, what must have happened to R2 star? Did it increase or decrease with activation? Is this rate slower or faster than this rate? Right. So the R2 star must have gone down. So R2 star A must be less than R2 star B, and that means delta R2 star, which is the difference between those, is less than 0. Okay. And so if we go through, if we plug these here, we're saying the difference between the active signal and the baseline signal divided by the baseline signal is sort of our measure of the percent change. Okay. And it turns out that we can approximate this with a first order Taylor approximation as minus the echo time times delta R2 star. Okay. And since this is negative, then typically this whole thing will be positive. All right. And so that's basically what the phenomenon we're looking at. It's that change in relaxation rate that we're measuring. And it turns out that, as we discussed before, that the main thing is that R2 star depends on how much deoxyhemoglobin you have in your brain. Okay. And so what's happening is, imagine this is a deoxyhemoglobin. This is a hemoglobin molecule. Okay. So this is in the lungs. Okay. So those red things are representing oxygen molecules that bind in the lungs. Okay. And then, so these are like the O2 molecules. Okay. And what happens is it delivers the oxygen up to your brain, and then your brain starts using up the oxygen, right? And so what that's going to do is that oxygen is going to get offloaded, and now you're going to be exposing these iron atoms here. All right, and that's the basic principle of FMRI, is that as those oxygen atoms come off, that shielding of the iron atoms goes away, and so now the iron atoms can perturb the field around them. Right? And that's this idea that if here, in this case, up here on the top, you sort of, all these atoms still have oxygen on them, okay? And so there's only deoxyhemoglobin, let's say, on this atom here, right? Okay, so that's going to be some amount of deoxyhemoglobin. Whereas here, all the oxygen has come off, and now you see all the iron atoms. And so that's going to lead to a lot more dephasing, and so therefore, we're going to have a smaller signal and higher R2 star. Okay. So given this picture and what we talked about, what happens during activation? Is there more or less deoxyhemoglobin in the brain when you're doing an activity on average? Okay, so we know that the signal goes up with activity in general. Okay. So, and given this picture and what we talked about, is this the active condition or is this the active condition? Okay, so we're saying this is the active condition. There's more deoxyhemoglobin in the brain. Okay, so if there's more deoxyhemoglobin in the brain than we expect during the active condition, the signal would have gone down, right? But we actually see empirically the signal goes up, right? So paradoxically, it turned out that this is the active condition for most of the brain. Okay, so actually, it's opposite what you might expect. So nature does not follow what we expect. It just does what it does. And it actually, on average, there's less deoxyhemoglobin in your brain when you're using your brain. Okay? So it turns out, why is that the case? So, this is, you could do a whole PhD thesis on that, and people have. So we're just going to give you the 2-minute version of it, which is there's competition between cerebral blood flow, which is how much blood's being delivered, right? So if I deliver more blood, I should have less deoxyhemoglobin, right? Because I'm just delivering more oxygen. So if I can increase CBF, then my deoxyhemoglobin should go down, right? Which would basically, if that was the only thing that was happening, then that would correspond with our picture of deoxyhemoglobin going down with activity. But the other thing that happens is there's something called CMRO2, which is oxygen metabolism. And that's what sort of we expect, is that the brain is using up oxygen, and therefore there's going to be more deoxyhemoglobin. And so there's these two competing factors. So CBF is delivering more oxygen, that makes deoxyhemoglobin go down, and then metabolism is using it up. Okay, so there's two competing factors. So if those were matched 1-to-1, if the increase in CBF, let's say the percent increase in CBF, let's call it percent change. So the percent change in blood flow divided by the percent change in CMRO2, if that was equal to 1, then they just cancel out. Okay, so then that would be like no bold signal. Okay, but it turns out that in practice it's closer to 2, and that gives us a bold signal. And so what that means is, because of over many years of evolution, the brain has figured out that when you do something, it better deliver more oxygen than it thinks it needs. And so there's almost a 2-to-1 or 3-to-1 ratio of delivery of oxygen over the demand. And so you always have, in general, more oxygen than you need, and therefore the bold signal goes up. All right, any questions on that? Okay. So let me, I think this will work. Hopefully this doesn't crash like it did last time. So in this example, we have the cerebral blood flow, which is bringing in oxygen through the arterioles into the capillary bed. And as there's metabolism, it is some of the oxyhemoglobin gets converted into deoxyhemoglobin. And so in the venules, or the small veins, we have both oxy and deoxyhemoglobin here. Now, with neural activity, we expect that metabolic rate will go up, and therefore we expect to have more deoxyhemoglobin. And that leads to more dephasing of the spins, and so actually there is going to be potentially initial dip, which we do sometimes observe. And so the bold signal may eventually go down, does initially go down. But what happens is fairly quickly the cerebral blood flow increases, and roughly by a 2-to-1 ratio compared to the CMRO2 increase. And so that delivers much more oxygen than is needed. And so overall, the amount of deoxyhemoglobin in the venules decreases, and therefore we have a decrease in deoxyhemoglobin, and therefore a positive bold signal because there's less dephasing of the spins. And then after the stimulus is over, or the neural activity is over, then gradually things return back to baseline. There can be this post-stimulus undershoot that is still being studied, but after some time things return back to their equilibrium state. Okay, let's give this a try. All right, so then an increase in the functional bold response will tend to increase the bold signal. Okay, so most people got that. So basically there's more O2 being delivered, therefore there's less DHB, there's less dephasing, and therefore the signal should go up. All right, so that's basically all you really need to think about is how much iron is going to be in the blood depending on what you do. All right. We're going to skip this one. We'll do this in the review session, okay? All right, so let's talk a little bit about the applications of functional MRI. So, I'm sure when whoever drew this picture did this, they were just joking, right? But it turns out that pretty much all those things that they've talked about have been studied with FMRI. All right. The only one I really couldn't find was donuts. But there's a lot of studies of like obesity, eating disorders, and what makes you crave food. So I think that covers the donuts. Everything else has actually been studied. Okay. And so there's probably I mean, there are definitely thousands and thousands of fMRI studies now already. And one of my favorite ones was this one from the group at Caltech, where this is an experiment where they're basically looking at willingness to pay. And so here what they did was they gave people the same wine in unmarked bottles. And in one case, they were told that the wine cost $10. In another case, they were told it cost $90. And they were asked to rank how good the wine was or how much they liked the wine. So if you look at this liking scale, when they're told that the wine costs $90, they're giving it this score. And then when they're told it costs $10, they give it this score here. Okay? And similarly, when it's 45 versus 5, you like the wine much better when it's 45 versus when it's 5. Okay? So this is why pricing is a big deal in business. Basically, oftentimes, sort of counterintuitively, sometimes you want to price your product much higher because that signals it's better, okay? And people are willing to pay. So like Apple gets away with this all the time, right? And so in this study, they were looking at the reward areas of the brain. And so here, let's just look at this one here where here is the brain response in that area as they're doing the 45 wine, and it sort of goes up, and it's sort of, as you would expect, it's sensing that there's more reward. Versus here for the 45 wine, it doesn't go up as much, and in fact, there's like even some negative response afterwards. And you sort of see the same thing between a lot of activation for the 90 wine, just a blip, and then this negative for the $10 wine. So from a reward point of view, how much you think something costs actually, even in your brain, whatever it's computing is actually computing that it's just not as good if it's a cheap wine. Okay? Yes?
Just out of curiosity, did they show the percentage of people in the study who's thinking followed the trend they were looking for?
That what?
Did they show the number or percentage of people whose thinking followed the trend that they expected?
Oh, you mean that didn't follow this?
Yeah, I mean, plenty of people probably would not care one way or another about the price, right?
Yeah, I don't know. I don't remember the details of the study, but I think, so that's a good question. There was probably some people who were like the cheap wine, That's why Trader Joe's does well, right? But I think in a blind taste test, then this was where everything was controlled. Okay. And also, in this case, they weren't actually, I'm not sure that the subjects were actually paying for the wine on their own. They were just told how much the wine cost. So all these things make a difference, right? If you're actually paying for it, then it's going to probably change the results. Okay, then you actually get a lot of satisfaction from that $10 wine because you're like, I saved a lot of money here, right? So it turns out that the details of the experiments actually matter a lot. Okay. I'm not going to play this movie, but you guys can do to the YouTube video. What this movie is, though, is that it's pretty cool work from Berkeley where basically they're trying to do mind reading, and so they play these people movies. And they've already mapped out what parts of the brain represent what visual images. And so what they find is that they can actually recreate the movies fairly well. And so I would recommend that you go look at this movie. It's not perfect yet, but it's scarily good in the sense that if I show you a movie and I just measure the signals from your brain, I can sort of tell what the movie looks like. Okay? So this is not at the point yet where anyone has to be worried that anyone is reading your mind, but maybe in 50 years that will be the case. All right. I think one of the groups is going to talk about real-time fMRI, and this is a case where by showing subjects how much activation they have in certain areas, it turns out that they can actually modulate the activity in that area, which is pretty amazing. So for example, if I told all of you to decrease your blood flow to your right insula, who here can do that? Yeah, maybe you could. I mean, if you were like a yogi, you probably could do it. But most of us couldn't do that. But it turns out that actually if you show people the signal in their brain, and so in this case, they were showing them the signal from this part of their brain, and then they were told to make the signal go up or down, that actually, just the way you learn anything, if you're given feedback, you can actually figure out how to modulate the signal in your brain. Okay? So this has been sort of, people have been trying this for the last 10 years, and one of the group projects will go into more depth on this. Okay, are there any questions before we leave that part. Okay, so the last thing I want to talk about is something called resting state fMRI. But to do that, we're going to first contrast it with task-related fMRI. So what we've talked about so far is what happens if you make the subject do something while they're lying in the MRI scanner. Okay. Resting state fMRI is sort of an interesting experiment. The experiment here is what happens if you just put someone in the scanner and just say don't do anything, and you just measure their brain. Okay. Now, you haven't told them what to do, and yet clearly their brain's probably doing something, right? We all know subjectively that our brains are always seem to be on. And so it turns out if you use fMRI to measure their brain, you might, and you look at one region of the brain, you might just end up with something that looks very much like noise. It's just going up and down sort of randomly. There's really no discernible pattern there. But it turns out that if you actually take that signal and ask what other parts of the brain have a signal that looks like that signal, there's actually a lot of structure in the signal. So for example, here in blue and green, we've got sort of the left and right motor cortices from someone just lying in the scanner. And you can sort of see those signals are very in sync. They're highly correlated. And in fact, if we took the signal from, say, the left motor cortex and asked what regions go up and down in a similar fashion, we can come up with these sort of correlation maps. Okay? And these maps look very similar to the maps on top, which were obtained when someone was actually tapping their fingers. Okay. So the observation was that the brain at rest is already functionally organized, and things that are going to be activated together when you're doing a task are already sort of talking to each other even when you're not doing a task. And this paper, the paper that launched this was published in 1995 by Biswal and others. And Bharat Biswal at the time was a graduate student at Medical College of Wisconsin trying to finish his PhD. He had this finding. It was rejected at least 3 times. So you can imagine, fairly depressing outcome. But finally his advisor was able to get it pushed through and published. And now this is one of the standard methods. So, and it took about 10 years after he published it for the field to finally come around and realize that this was real. But at first everyone thought that this was just noise, it was just an artifact, it couldn't be real, this couldn't exist. So sometimes when you do something really new, there's going to be a lot of opposition to what you're doing. Okay. But after about 10 years, people started realizing it was real and that it was reproducible. And in fact, there are many, many networks in the brain. And so this is just showing 6 networks that people have found, including visual, motor, and the one we're going to talk about a little more is the default mode network. Okay. So this is a network in your brain which is pretty much centered on this back area of your brain here, a few centimeters down, called your posterior cingulate cortex. So it's also shown here. And in red here, you're just seeing all the areas that are synced with that part of the brain. And that's also shown in these time courses going up and down. And then, interestingly enough, there turns out to be another network here in blue, which if you look at, which has these time signals here in blue, and you can sort of see they're anti-correlated. When one goes up, the other goes down. And this is happening all the time in all of us, that we have these 2 major networks where the default mode network is really active when you're sort of thinking about yourself. And then as you start to do something that requires attention, it goes offline a little bit, and the attention network comes in. And so for all of us, typically we're going back and forth between these 2, sort of the internal and external views of the world. And so there's been a lot of interest in this and quite a lot of literature discussing how this might be impaired, for example, in depression, where you might expect the default mode network to be hyperactive. And so this is one of the major networks that has been identified using fMRI. And so right now, for example, there's many large projects, including the Human Connectome project, which use MRI. And so they acquire very nice anatomy. And they also acquire, use fMRI. This is done with fMRI. And it's done with resting state, so resting state RS fMRI, where by just laying people in the scanner for 10 or 15 minutes, they can actually start mapping out all these connections in the brain. Okay? The last one we'll talk about, probably not today, but on Wednesday, is diffusion imaging, which is what we use to measure the wiring in the brain. So the amazing thing, if you think about it, it's very commonplace, so sometimes I forget how amazing this is, but the amazing thing is you can put someone in the scanner for 30 minutes or an hour, just put them in there and come up with a map of their brain. You can basically know their wiring and also how the different areas are connected to each other. Okay? And plus you have a nice picture of their brain. So I think this is sort of one of those things that is definitely going to be one of those steps, is already one of those steps that's allowing us to understand the brain much better. So before we switch for the last 10 minutes, are there any questions? Okay. Someone could have told me to get rid of that. Okay. All right. Okay, so let me just talk about, we're going to spend the last 10 minutes just setting up some stuff. Just to give you a sense of where we're going, Wednesday there'll be one more lecture on covering moving spins and also RF pulses. Professor McVeigh will then give a lecture next Monday, sort of summing things up, and then the quiz will be next Wednesday. Okay, so we're getting towards the end of MRI. So what I want to talk about for about 10 minutes here is what happens when we have moving spins. All right, so far we've mostly talked about what happens when the spins are static and we can create images of them. And so it turns out that this concept of phase is going to be really important. So we've already talked about phase in the context of creating images, but now we want to talk about how can you use phase to measure velocity. Okay, and so the idea is this. Let's say we have spin A and spin B. Okay, and spin A is going to be static, and spin B is going to move. So we let that spin B is moving in the presence of this gradient field. So this is the gradient field. And so you can sort of see spin A is static, and spin B moves to the right, okay, in the positive x direction. And it's going to an area where so they both experience a gradient field, and in this case it's a negative field, okay. So they're going to spin slower with the reference to isocenter. So this is going to lag behind by about 90 degrees, right? So it's going to go counterclockwise by 90 because it's slower as opposed to spin going clockwise. But spin B moves into an even stronger part of the field, and so it's going to lag behind by even more, 135 degrees. Okay? So there's this differential lag between those two spins. Now what we do, and so let's say we wait some amount time t for that to happen. Now we're going to reverse the gradient and wait an equal amount time t. Okay? So spin A experienced a negative gradient and then a positive gradient. Okay? And it's experiencing exactly the same gradient because it didn't move. Right? So what's the area of that gradient? 0. So it's still in the same place in K space, and in fact, this 90 degree that it lost, it totally recovers it. So it goes back, and then it recovers it. Okay, so that's what spin A experienced. Spin B is experiencing something like this. It's moving into a stronger part of the gradient, right, over time, and then we flip it, and it's even moving in an even more stronger part of the gradient. Okay, so it's going into these it's feeling different gradient fields. And so what happens is, since it keeps moving this way, it moves out into an even stronger part of the field, which has now been flipped in sign, so it overcompensates. So it went back 135. Ideally, we would have just brought it back 135. Because it's moving into a stronger part of the field, it sort of goes past 0. Okay. So now it has a net phase. Okay. And basically, it turns out that net phase it accumulated is proportional to the velocity. Okay. So the amazing thing is that the phase of the spins tells you about how fast they're moving. Okay. If you apply the sort of gradients. Okay. So let's take a look at the expression for the phase of a moving spin. So once again, we have that phase is simply the integral of how much incremental frequency we have. That's just given by gamma times the delta B field. And as we talked about in imaging, that's just gamma times the dot product of the gradient with precision vector. And previously, this was static before. Okay, so it was just R. Now we're going to give it some time dependence. So that's the only thing we're changing. So now these all have time dependence. Okay, and so we're just going to go through and look to what happens when that has time dependence. So we're going to consider a very simple motion of a spin with some velocity and acceleration. Okay, so assuming this was the initial position, then at any point in time, you can just go back to your basic physics and add the velocity times time plus 1/2 at squared. And when you do that, you simply plug that into this expression, and then you do these integrals. You find out that there are 3 different integrals for each of these components. One that's related to what's the 0th order moment, the 1st order moment, and the 2nd order moment. Right? So we're really going to just focus on this one here. Okay. For now, we're going to ignore this for now. All right. So that's just shown here. So the 0th order moment is just the area. Okay, and we're going to use these bipolar pulses like this. So that has, typically we're going to use pulses that have area of 0. So we're going to make that go to 0. Okay, so this pulse has no sensitivity to where you are in space. And we're going to make it to the 1st order moment actually is going to have some sensitivity, and that's the sensitivity to velocity. So you can see that 1st order moment is just the gradient times time, the integral of that. Right. So previously in imaging, we've only looked at the areas of gradient. So we spent all this time looking at the area of the gradient. Now what we're going to do is we're going to look at the 1st moment of the gradient, because that's what we need to sensitize ourselves to velocity. Okay. So once again, this is the bipolar pulse that we just talked about. So it goes down for a certain amount of time and then up for a certain amount of time. 0th order moment is 0. And it turns out, if you do the algebra, the 1st order moment of this is g0 t squared. So it's very similar to physics when you look at the moments of beams and things like that. Okay. So one simple way you can tell if something has a 1st order moment is if you imagine this is a function of time, but let's say this was just, if we thought about these as, instead of a waveform over time, as like concrete blocks, where one is pulling down and one's pulling up. So you can imagine this has some force going this way, and this has some force going that way, right? Depending on its area, right? And that's going to cause a rotation of that beam, right? If this was some beam, and I applied forces like that, so anything that sort of causes that sort of rotation is going to have a 1st order moment, right? So this leads to a method called phase contrast angiography, and we'll just end with an example of that. Where essentially I can apply a pulse with the gradients like this, and this has some 1st order moment, so it's going to give me some phase term phi1. When I reverse it, the 1st order moment just changes to the negative of that, and so that gives me some other phi2. And both of these depend on the gradients and how long they're on, and also the velocity. Okay? And in this case, if I take the difference of those two, phi1 minus phi2, I get an expression that is related to velocity. So the experiment is I measure the change in phase, I know the gradient, I know the time it's on, and I just solve for Vx. All right, and that's the phase contrast angiography experiment. So here's an example of that, where we're looking at, once again, these are now the vessels coming out of the circle of Willis. So some anatomy, you have your carotids and your vertebrals going up here, and then there's a little circle there named after this guy named Willis, which sort of balances the flow, and then your other arteries come off of that and supply the rest of the brain. So that actually is a really important area of the brain. If that gets screwed up, the blood flow can be impaired quite a bit. So what we're looking at here is, and one thing about it is we can, depending on which way we apply the gradients, we sensitize ourselves to different flow. So in this case, we want to look at the flow here on the left, the flow moving in this direction. So that's called anterior to posterior flow. And so you can sort of see here that we're sensitized to that flow there. Okay? And so in that case, we would have, let's say this was the the y direction, we would apply gradients in the y direction to sensitize ourselves to flow in that direction. Let's say this is the x direction. In this case, we want to sensitize ourselves to flow going left to right. Okay, so we apply a gradient in this direction, and that's going to sensitize ourselves to flow. And so you can sort of see here that we have positive flow on this side and negative flow on that side. And that's good because that's how it should work. The blood comes up, some goes this way and some goes that way. Okay. So phase contrast angiography is one of those standard methods that we use to sort of diagnose blood flow in the brain, especially in the big arteries. And the other example here is of looking at blood flow in the heart. And so since this is the end of the lecture, I'll just go ahead and play this so it doesn't matter if it stops, okay? So we'll just go ahead and try this one more time.